In the last class, we have seen up to the epigenetic changes. So actually, we have split that section to the two epigenetic changes we have already covered. One is the histone deacylation and histone acylation and deacylation that we have already covered. The effect of or the influence of the carcinogens. Then DNA methylation and how that is actually producing the carcinogenesis. Now we will go to the third important epigenetic changes. Again, the epigenetic changes may not be that much familiar to you, many of you. So the first one, the third one, and again one of the important among the epigenetic changes, the microRNA. So many of you might have heard of the microRNA. For those who are not familiar with that one, because in the textbook that is not being described. So the microRNA is actually synthesized as the primary miRNA or primary microRNA by the RNA polymerase 2, and it is processed within the nucleus as with the help of an enzyme named DROSHA, which is the RNase 3 enzyme, which process the primary microRNA into a 70 nucleotide precursor, that is pre-miRNA. So these two are different. The primary miRNA is processed to the pre-miRNA. The pre-miRNA, so the primary is converted to the pre by the help of the enzyme DROSHA. This happens within the nucleus. And once it is in the nucleus, it is transported from the nucleus to outside, because the site of action of this microRNA is in the messenger RNA. Okay, and so it has to be the messenger RNA usually leaves the nucleus through the nuclear pores and then reaches the ribosome. So likewise, the pre-miRNA also has to leave the nucleus with the help of a protein that is known as the exporting protein. Now, once it is in the cytoplasm, there is another enzyme which is known as dicer. So this dicer is a RNA polymer RNase 3 enzyme which cleaves the pre-miRNA into the miRNA. So primary RNA, pre-miRNA, and miRNA. So this is the pathway through which it is undergoing. And what happens is that this even the double-stranded the pre-miRNA, when it is converted to, or the primary mRNA, pre-mRNA is actually a double-stranded RNA. Okay, and this double-stranded RNA itself is having complementary strands, so it cannot attach to anything else. So what happens is that inside the cytoplasm, the guide strand is separated from the other one, and this guide strand will combine with another substance in the cytoplasm which is known as RISC, or the RNA induced silencing complex. Okay, this RISC will then again come to the messenger mRNA. Okay, and it will attach to the mRNA. And when it is attaching to the miRNA, what happens is that this mRNA will, so this is the mRNA. This mRNA will undergo degradation. I have given it in the diagrammatic way. So this is the polymerase enzyme which will synthesize the microRNA. So this is the gene for the microRNA which will synthesize the primary mRNA. And this primary RNA, with the help of the enzyme DROSHA, will be converted to pre-miRNA. The exporting will transport it outside the one, and it will have a stem loop. This pre-mi up to the pre-miRNA microRNA, it will have a stem loop, and that stem loop will be cut by the enzyme dicer. And this dicer again will separate the strands into two single-stranded one. And the leading strand will combine with the RISC, RNA-induced silencing complex, and this will get translocated into the target mRNA. So this mRNA and miRNA will combine together, and this will make the mRNA into a double-stranded structure. And this double-stranded structure disintegrates the integrity of the mRNA, and mRNA will be converted to, or the fate of such mRNA, there are two fates for such an mRNA. Either the mRNA will undergo degradation, or what happens is that when it is attaching to the ribosome, it cannot enter into the ribosome. So it will create a truncated or an incomplete protein. So such a truncated or incomplete protein cannot function as a enzyme or as a receptor. So that will stop working. So this is the principle of the microRNA and how the microRNA is producing the inhibition of the protein synthesis. Now, the mechanisms of the carcinogenicity by mRNA or microRNA changes. First one is the epigenetic modification. So first one is the epigenetic modification, and the one that is producing such epigenetic modification by mRNA is arsenic. Second one is the activation of transcription factors. The transcription factors that are involved in this process, same picture, but I have mentioned the 7 different mechanisms which are acting on this miRNA induced pathway. So this is not actually there in the textbook. I am explaining this, or I created this slide to make you understand these things. Okay. Now, activation of the transcription factors will be produced by another carcinogen, that is a benzopyrene. Then disruption of miRNA biosynthesis is inhibited by the arsenic. Then oxidative and cellular signaling pathway is inhibited by another carcinogenic substance, that is a cadmium. Then nuclear receptor mediated regulation is done by the phenobarbital. Then feedback regulation from the target gene is produced by the TCDD, and the tissue and chemical specific changes will be induced by the bisphenol. So these are the changes that is produced by the each carcinogens, which is mediated by the microRNA changes. Okay, so the major epigenetic modifications that will produce carcinogen, that will end up with the carcinogens. First is the epigenetic modification. We have already seen the epigenetic modification, how the DNA methylation, histone modification, or mRNA. So these 3 things we have already seen. As an example, as I already mentioned, is the arsenic, and the arsenic exposure will lead to the hypermethylation of the tumor suppressor. So you can see it is produced by multiple mechanisms. Arsenic produces epigenetic changes by multiple mechanisms. One is hypermethylation of the tumor suppressor gene. We have already seen how the hypermethylation produces inhibition of the gene transcription. So now what happens is that when the tumor suppressor mRNA or tumor suppressor mRNA, the promoters, when they that will lead to the reduced expression of the tumor suppressor. So suppressors are reduced. What will happen is that initiation and progression will be enhanced. So this actually contributes to the epithelial to mesenchymal transition. So many times what happens is that in certain carcinogenesis, the epithelial cells are converted to the mesenchymal transition, and that mesenchymal cells are responsible for the promotion of the cancer. So that is one mechanism how the epigenetic modification is producing carcinogenesis. Second one is the activation of the transcription factors. Many of the agents, for example, the benzopyrene, is having the ability to activate the transcription factors such as P53, NF-kappa B, MYC gene, or AHR. In this AHR, we have already seen. These factors actually bind to the promoter regions of the microRNA genes, and that will alter the transcription of the microRNA genes. Then, disruption of the miRNA biogenesis. This the DROSHA, we have already seen the role of DROSHA, exportin 5, and dicer in the formation of mRNA. So what happens is that there are certain carcinogens which are capable of inhibiting or degrading the already formed proteins like the DROSA, dicer, and the exportin, or they may affect the transcription of the, or they will downregulate the transcription of these enzymes. So what will happen? The total mRNA, microRNA will be reduced. So when the microRNAs are reduced, the complex or the RNA mediated inhibition, which is otherwise known as RNAi or RNA inhibition. So RNA inhibition of the carcinogenic genes will be reduced when the RNA inhibition is suppressed. In case of a procarcinogenic gene, that will initiate the development of cancer. Okay, the next one is the oxidative stress and signaling pathway activation. So oxidative stress we will see later in details in this session itself. So the chemicals can induce the reactive oxygen species that will activate the stress pathways. We will see these pathways later. And they will downregulate or upregulate certain specific miRNA. Okay, the examples are the miRNA 21 and miRNA 34A in case of cadmium. And this cadmium generate ROS and activate the MAP kinase pathway. And this lead to the overexpression of the MIR21, which is associated with the anti-apoptotic effect. So when the when it is exerting the when this gene is activated, or when this microRNA gene is activated, what will happen? That will produce more anti-apoptotic effect. That is, even when the defective cells are formed, those cells are not dead, which is a classical change that is occurring in the progression stage of the cancer, tumorigenesis. The next one is nuclear receptor mediated regulation. The nuclear receptor regulations, we have already seen this significance of the CAR and PXR, estrogen receptor, aryl hydrocarbon receptor, all this. And these receptors are capable of acting as the transcript. So they are known as the nuclear transcriptors. And what happens is that when an agonist is binding with these receptors, they will get translocated into the nucleus. Once they are entering into the nucleus, what happens is that it increases the transcription of certain genes that are present in the DNA. Okay, so these receptors can upregulate and downregulate specific miRNA in the target genes. So this is one of the mechanism which is most commonly seen in case of the breast cancers. Then phenobarbital. The phenobarbital activates the CAR, and the CAR alters the expression of these miRNAs, promoting a liver growth and possible tumorigenesis in the liver. So that will end up in the hepatocellular carcinoma. So this is how the phenobarbital, for on a prolonged dose and prolonged use, it is producing the hepatocellular carcinoma. Next is the sixth mechanism out of the 7 is the feedback regulation from the target gene changes. Now, when the mRNA targets are upregulated by chemicals, the miRNA levels may adjust through a feedback loop. That is, when a particular miRNA, say, imagine that 100 copies generally the basal transcription level. Now, when there are certain activations in the DNA, what happens is that the transcriptional activation can result in 200 mRNA. Okay, when the level of mRNA in the cell increases, there is a feedback mechanism. Okay, there will be a feedback. So this feedback will lead to increased level of miRNA. Okay. This increased level of miRNA will inhibit some of this mRNA. So if there are 100 miRNA, 100 miRNA, what happens is that out of this 200 produced, the 100 only will be activated. So even when the expression of miRNA, so this feedback is actually a protective mechanism to provide a constant number even when there is transcriptional activation. Hello? Hello? Someone trying to ask any questions?
Yes, sir. No. Participants, can you
Okay.
please mute yourself? You can continue your presentation.
Okay. Okay. So the classical example of such a feedback regulation is with the dioxin. So dioxin is actually increases the responsiveness of the mRNA of many genes, and that will increase the level of miRNA by this feedback mechanism. Okay, and when this feedback mechanism is activated, AHR receptor is more concerned with the lipid metabolism, especially the steroidal mechanisms, inflammation and carcinogenesis. So when the levels are increased, what will happen is that the chances or the promotion of this normal cells to a tumor cell, the chances will be elevated. Okay, so this is how one of the mechanism of the TCDD. And I have to say at this point that a carcinogen may not have only one single mechanism. It will have multiple mechanisms that will contribute to the generally one substance that is capable of producing a particular cancer will not be acting through a single pathway. It will be it will be a classical carcinogen when it is producing the carcinogenesis at multiple pathways. Otherwise, just it is a toxic material. It may not act as a carcinogenic material, because the carcinogenic materials will produce carcinogenesis by or by undergoing so many changes, carcinogenic changes. Even for the initiation, progression and propagations, it will require multiple pathways. Okay, the next one is the tissue and chemical specific effect. Now, coming to the tissue specific effect, the miRNA is specific to certain tissues. Not all the miRNAs are present in all the tissues. In all the tissues, for example, in case of bisphenol, it can produce the toxic effect at very specific organs, because it can alter the miRNA expression related to lipid metabolism in liver, whereas it produces a neurodevelopmental detrimental effect on the neurodevelopment in case of brain, and in ovary it influences the reproductive hormones that is related to microRNAs. So a single chemical can produce multiple mRNA inhibitions, the miRNA inhibitions at multiple organs. That is the reason why these agents generally, even when they can produce mRNA inhibition, they are specific to certain organs. And in case of miRNA and chemical carcinogen, this is what is given in the textbook. I have already explained these things. And the miRNA can act as the tumor suppressor genes, tumor suppressor agents, or oncogenes. Okay, so when they are acting as a tumor suppressant, what happens is that the miRNA, increased miRNA, they will suppress the anti-cancer genes or the anti-carcinogenic genes. When they are acting as oncogenes, they will inhibit the or the tumor suppressor genes. Okay, and this such miRNAs are known as onco-miRNAs. Okay, examples of this onco-miRNA. These are the examples of the onco-miRNA genes, and this onco-miRNA upregulation is associated with environmental carcinogen exposure. The typical example, as we have seen in case of the bisphenol and other agents, and the carcinogenic effect may or depend on the balance between the tumor suppressor and onco-miRNAs. So this is actually this miRNAs are the inhibitors of tumor formation, whereas the onco-miRNAs are responsible for the development of the tumor, the initiation and the progression of the tumors. And in the body, there exists a balance between these two miRNAs. And when this tumor suppressor miRNAs go down, or when these oncogenes are going up, then that will result in the process of carcinogenesis. Okay, and this miRNA profile is another method where we assess the possibility of the carcinogenicity, because they are the indicators of the carcinogenic process. They are the biomarkers of carcinogenic exposure, and they are tools for identifying the potential chemical carcinogens. For example, the long-term exposure treatment with the phenobarbital or the phthalate induces this miRNAs. So when this miRNAs are elevated, that indicate that the carcinogenesis of phthalate or phenobarbital is about to come or about to occur. Okay, so that is what you call as the biomarkers. When their levels are elevated, so they are of diagnostic significance as well as experimental significance when you go for the long-term exposure studies. So this is the pictorial representation as given in the cataract handout. This is the miRNA levels in the normal tissues, and miRNA function as suppressor, tumor suppressor genes. So what happens is that they will suppress the oncogenes. The transcription of the oncogenes will be suppressed. So this will have an anti-tumor effect. Okay, such miRNA will have an anti-tumor effect. While this miRNA, when they are function as oncogenes, they are pro-carcinogenic miRNA. So such two types of actually I previously mentioned, there exists a balance between these two anti-tumor and the pro-carcinogenic mRNA. And when this is disturbed, that is when the anti-tumor is decreased and pro-carcinogenic miRNAs are elevated, that will lead to the development of the tumor or the carcinogenesis process. Now, coming to the immunosuppression, the next one is with this we finish the epigenetic changes that are associated with the carcinogenesis. Now comes the immunosuppression and carcinogenesis. Again, immunosuppression is a very significant thing in the carcinogenesis. And the immune surveillance is a critical defense mechanism that detect and eliminate early pre-neoplastic cells. Because when there is alteration in the DNA, sometimes what happens is that the antigenic epitopes on the surface of the cell, okay, when the DNA changes, okay, due to mutation or some other effect, or when the carcinogenesis, the nucleus is the first thing that is affected. Now, this change in the DNA can change the epitopes on the antigenic epitopes on the surface of the cell. Okay, when this is altered, what will happen? The cell itself will recognize it as a foreign cell inside the body, and this foreign cells or this foreign or mutated surface protein can activate the immune response, especially the T cell mediated immunity. So the T cells will be activated, the complement system will be activated, the antigen presenting cells APCs will be activated, and the cells will be destroyed. Okay, so immune surveillance is one thing that is around the clock running in the body as a mechanism to inhibit the already promoted cells. Okay, so that is why even though when there are a lot of mutations that is happening inside the body, it doesn't develop into a tumor in the body. Now, what happens is that imagine a condition where the immune system is inactivated. So what will happen? This mechanism of detection will be inhibited. So this mechanism do not exist in the body, or it will be existing at a very low level in the body. So what will happen? The already promoted cells. Promoted cells means the cells that are promoted to a stage of carcinogens. So it has to undergo the progression in the next stage. So the progression is actually prevented by the immune system. And when the immune system is compromised, what will happen? The cells that are selected as the clonal cells of the tumor, they will be expanding, or what you call as the clonal expansion. So the clonal expansion will be activated when the immune system is activated. Okay, so that is even the evidence was selected from the organ transplant patients. So organ transplant patients, to prevent the graft reject, they have to continuously take, or lifelong they have to take immunosuppressant drugs like the azathioprine or the endoxan or some drugs like that, immunosuppressant drugs like that. So in those patients who receive the immunosuppressant agents, or in certain disease conditions like HIV, where the immune system is impaired, the chances of carcinogenesis is more. The reason why in case of immunosuppressive genes and the in case of immunocompromising diseases like HIV is the surveillance by the that critical defense mechanism exerted by uh, the immune system is impaired. So the pre-neoplastic cells we can freely convert it to the neoplastic cells. There will be clonal expansion, progression, and developing into a mature cancer. Okay, and there are certain tumors which is working in the opposite way also. They can invade the immune cells. Once it is developed, they will impair the immune system. Okay, especially this is seen in case of the fluid tumors like the leukemia or the tumors that are associated with the bone marrow. In all these cases, they will suppress the immune system. So that is another, or that is known as immune invasion. They will affect the cells that are responsible for such a surveillance, and that surveillance is inhibited, and that inhibition of that survival will produce or convert the pre-neoplastic cells to the neoplastic cells. Okay, and again, non-genotoxic carcinogen may also act through the immune suppression. So this is the mechanism how the immunosuppressant drugs like the azathioprine or the cyclophosphamide, such drugs are capable of producing or they increases the chance of development of cancer. Okay, they do not affect the they do not produce any genotoxicity. There will not be any mutation or there will not be any changes in the DNA, but they can activate the or the suppress the immune system, and that suppression of immune system will actually activate the tumor progression. Okay, then apart from the immunosuppressant drugs, the bisphenol, atrazine, phthalate, PDB all are responsible for development of the carcinogenesis by this mechanism. But these agents do have some effect on the nucleus also. Okay, or there can be some changes in the DNA. Okay, the classical immunosuppressant drugs, they will not affect the DNA. Generally, they will not affect. So some effect will be there in case of DNA, but they will not produce any genotoxicity or mutational effect. But this bisphenol, atrazine, and phthalates, they are capable of modifying certain things that is happening in. So the phthalates and bisphenol we have already seen by affecting the miRNA, microRNA, they can produce tumors. Then the next important reason for the carcinogenesis is the ROS, or the reactive oxygen species. Okay, reactive oxygen species, all of you know, you might have heard about it because anti-oxidant drugs are a hot area of research, not only in carcinogenesis, but in the implication of so many other lifestyle diseases also. Okay, the common ROS include the superoxide anion, hydroperoxyl radical, hydrogen peroxide, and hydroxyl radical. And these are the intracellular ROS pathways. This is actually taken from the Castrate and Dow. The oxygen, when it is combining with the one electron, it becomes the superoxide, that is CO2, O2 minus anion, and this O2 minus anion will combine with water to form hydroperoxide radical, and this hydroperoxide radical can combine with an electron and a hydrogen to produce the hydrogen peroxide molecule. Okay, so at this step is the place where the SOD or superoxide dismutase is acting. This is the place where the catalase is acting. Catalase is producing, and because superoxide is actually neutralized by the superoxide dismutase enzyme, the catalase is inhibited by the, or the catalase is converted, or the catalase is capable of converting the hydrogen peroxide to water and molecular oxygen. And this H2O2 itself can propagate this, or it goes as a chain reaction. When this H2O2 will combine with an electron, that will produce two molecules of the hydroxyl radical, one OH minus, and that is the hydroxide anion, and there is hydroxide radical also. So all these agents are highly active reactive oxygens, and the cellular oxidants, this change is happening inside the mitochondria. So generation of ROS is an essential thing that is required in the body. So it is not a toxic process. So the mitochondria to work, it will require the mitochondrial membrane potential. You might have heard of the MMP, mitochondrial membrane potential. So this mitochondrial membrane potential is required for the synthesis of ATP. From ADP, one phosphate is added by the ATP synthase enzyme, and the ATP synthase is actually you might be remembering how it is working with the 120-degree rotation. With each rotation, it is phosphorylating the ADP to ATP. So this repulsion actually requires the membrane potential, mitochondrial membrane potential. Then the second one is the cytochrome. Cytochrome also for the energy production, it will require the reactive oxygen species. So reactive oxygen species is something that is really required for the body, but when it is exceeding a particular limit or when that balance is disturbed, that will lead to the development of the internally, the third pathway where the this is happening is in the inflammatory cells, and this is one of the major thing that is required for the inactivation of the organisms that are entering, like the virus, bacteria, by the macrophages, neutrophils. All these cells will produce this type of reactive oxygen intermediates, and this reactive oxygen intermediates will damage the membrane of the bacteria, virus, or fungus, or whatever the organism, invading organism. So that is then peroxisome. So when we studied about the PPAR, the peroxisome proliferation activation receptors, we have studied the role of the peroxisomes. So these are responsible for the maintenance. Even the normal functioning of the cells also will require the development or the generation of ROI. Now, this is the one actually really that is responsible for the generation of ROIs, especially in case of chemical carcinogenesis. Then first one is the redox cycling compounds. That is the oxide anions. Then metals like the iron, they are responsible for conversion of this H2O2 to OH minus and the hydroperoxide radical. And this hydroperoxide radical, so the iron becomes Fe2 plus and Fe3 plus. This reaction is known as the Fenton reaction. Fenton reaction again is a normal thing that is happening within the cytochrome. That is a normal thing that is happening in the cytochrome, but when it is happening outside the cytochrome, that becomes more toxic, especially when it is happening near to the nucleus. That will produce a lot of the hydroperoxide radicals, and the radiation injury, especially the beta and gamma radiations, they are capable of producing hydroperoxide radicals, and these hydroperoxide radicals are responsible for the development of carcinogenesis. Then the cellular antioxidants. Already, all of you might have heard of these antioxidant mechanisms. There are two classifications. One is the enzymatic, and second one is the non-enzymatic. So the enzymatic will include SOD catalase, glutathione peroxidase, then glutaredoxin, thioredoxin, all those agents. The non-enzymatic will include the vitamin E, glutathione, vitamin C, catechins. All these are responsible for the antioxidant mechanism. Now, really the problem happens when the oxidant levels are more than the antioxidant levels that is present in the body. And this will produce oxidative damage in the DNA, RNA, lipid, and protein. This we have already seen in the previous sessions, how they are capable of producing adducts in the DNA and how the mutagenesis is produced. Okay, that we have already seen in much detail in the earlier session. Then the sources, antioxidant defense mechanisms, we already discussed these things. This also we have seen in detail. And the immune system, when I mentioned about this, there is a specific term called as respiratory burst. Respiratory burst actually generates the ROS. So don't think that it is associated with the respiration in the body. It is associated, that is happening increased level of oxidation that is happening in the body, in the cell, within the cell. Okay, and this is the normal thing that is happening in any defensive cell, like the Kupffer cells or the macrophages. So this is a normal thing that is happening. Okay, and those cells definitely have the protective mechanisms for the DNA also within the nucleus. They have the protective levels of high levels of protection against the oxidative injury. Okay, the chemical induced ROS. This we have already mentioned in brief. So we will see the three mechanisms how the cytochrome P450 enzymes generate the reactive oxygen species. The first one is called as the redox cycling. Okay, in the textbook they have mentioned only this name. So I'm giving the mechanism as well as the examples also. The examples are not given in the textbook, but these are the examples for this mechanism. The first one is the redox cycling. Redox cycling means there will be a repeated oxidation and reduction that is happening in the cell. So that will produce a reactive oxygen species, or there will be a fluctuation in the reactive oxygen species. So it will go up like this. There will be increased spikes of oxidative stress that is happening inside the cells. So this is the mechanism of the carcinogenesis by the paraquat and quinolone. Then the next one is the peroxidase catalyzed oxidations. So peroxidase enzymes use the hydrogen peroxide to damage the peroxide to oxidize the substrate. So we have already seen the peroxidase is responsible for converting the water to hydrogen. Water to hydrogen peroxide. And the molecules like benzidine, they are capable of activating the peroxidases. Okay, so this benzidine test actually or the peroxidase test, we actually used to detect the or what you call as the benzidine test for the detection of hemoglobin. The same thing is happening in vivo also. The third mechanism is called as the futile cycling. Futile cycling means a non-functional cycling. It usually happens with the nitrobenzene and aflatoxin B1. So what happens there is that it will undergo the same compound will undergo auto oxidation and reduction. Oxidation and reduction. So this will go on cycling. So for this oxidation and reduction, the cells will require the NADPH for the energy generation. And such thing, when it is continuously shuttling within the oxidative and reduced state, what happens is that the cell become deprived of the oxygen. And when the cell continuously undergo hypoxia, the chances of developing or the cell for converting to a tumor cell, the chances are more, because the tumor cells are always microaerophilic. They are the cells which are capable of surviving at a very low oxygen tension. Okay, so the chances of developing it, especially when highly functional cells are converted to a microaerophilic condition, that can lead to the development of or that can lead to the initiation of cells of the carcinogen or initiation of the cells with the carcinogenic properties. So that is the significance of the futile cycling. Now the chemicals that increase ROS, these are the chemicals that activate the ROS system by the mechanisms. Then peroxisome proliferation and ROS. So I already mentioned the PPR activation, PPR alpha activation, or the peroxisome proliferation. We have already seen, which is one of the classical pathway by which the phthalate and clofibrate they will induce the cytochrome 4A subunits, and that will increase in peroxidase level, which will lead to more hydrogen peroxide and increased oxidative stress. Okay, and there is a strong link actually discovered between the peroxisome proliferation as well as the ROS generation and carcinogenesis. So this is one of the proposed mechanism of peroxisome proliferation induced by the phthalate and clofibrate. And as I already mentioned, this is not the sole mechanism by which they produce carcinogens. They, as I mentioned previously, these carcinogens or the classical carcinogens, they will produce carcinogenesis by multiple pathways. Okay, so you cannot say which is the major pathway, which is the minor pathway. Every pathway that is favoring or converting the cells to a carcinogenic or pro-carcinogenic or pro-onco cells. The next one is oxidative DNA damage and carcinogenesis. The reactive oxygen species, there will be unbalanced ROS can lead to the macromolecules, and the ROS, this already I have mentioned how the reactive oxygen intermediates are capable of producing uh, a change in the DNA that I already mentioned in details how the attacks are formed. Then the modifications that is happening in the purine pyruvate bases, as well as the how they are capable of destroying the or damaging the DNA cross-linkings. Now, the consequence of persistent DNA damage will lead to the transcriptional arrest or activation. Then there will be signal transduction pathway induction, then replication errors, and that will lead to the genomic instability. Or these four pathways together or singly is capable of producing a carcinogenesis. Then example is with the hydroxydeoxyguanosine, how it is producing the carcinogenesis. Then the next one is the mitochondrial DNA susceptibility. Now, this has been coming up as a major or one of the pathways by which the carcinogenesis happens. So, when you compare the DNA that is present in the cell with the mitochondrial DNA that is uh, present in the cell, the mitochondrial DNA is much more vulnerable to oxidative stress or the oxidative damage, because there are three reasons. First one is proximity to the ROS generating. The mitochondria itself is the one that is generating ROS system. So unlike the nucleus, which is far away from the site where the reactive oxygens are produced, these are capable of producing or they are very much close to the uh, this nucleus, and the lack of histone protection. So the generally the DNA will be wound around the histone protection, histone protein in case of nucleus, but in mitochondrial DNA there is no histone. They are free DNA only. So the oxidative agents can act on any site. And the third important mechanism is that the nucleus do have the DNA repair capacity. We have studied the single nucleotide excision repair, then base excision repair, then sister chromatic exchange, all those things, the mechanism by which the DNA will be surviving a mutation. Okay, but that survival mechanism or that repair mechanism is not present in the mitochondria. So because of these three reasons, mitochondrial DNA are much more sensitive to the carcinogenic attack. Okay, so there is high mutation rate in DNA. The reactive oxygen species overproduction will be there. Then impaired oxidative phosphorylation. Okay, so this impaired oxidative phosphorylation, especially the agents which, yeah.
It is 8:45 now. Can we go for 5 minutes break, sir?
One more slide.
Okay, sir.
Okay.
Okay.
So the impaired oxidative phosphorylation. Then the next one is apoptotic evasion. Okay, the apoptosis that happens in the mitochondria, irrespective of the cell or even the mitochondria can initiate the internal apoptotic pathway, but the mitochondria sometimes survive the apoptotic pathways, and that will produce multiple mitochondria which are already defective. Second one is mitochondrial biogenesis. Unlike any other cellular organism, mitochondria reproduce within the cell independent of the cell cycle. All other organelles in the cells are replicating only at the time of the mitosis, but mitochondria will undergo at any time irrespective of the cell cycle. Then release of pro-inflammatory factors. The mitochondrial damage can lead to mitochondrial DNA damage can lead to the mitochondrial DAMV, that is the mitochondrial DNA fragments, which will trigger the chronic inflammation in the cell. And the next one is the mitochondrial and the nuclear communications. So mitochondria actually communicates with the nucleus regarding the energy levels that is to be produced. So the nucleus actually controls the mitochondria, how much energy need to be produced. But when this crosstalk is inhibited by the DNA damage that is happening in the mitochondria, because the mitochondrial DNA is under the control of the nuclear DNA. So when that crosstalk is lost, that will lead to the cell proliferation as well as the survival, because the mitochondrial replication and the mitochondrial activity will not happen in normal cases. Okay, now these are the mitochondrial mutations. These are the agents that will produce mitochondrial mutations, and thus uh, subsidiary pathway in case of the. Okay. Now, coming to the next one, that is ROS-induced lipid peroxidation. So the same way, it produces nuclear attack. It is capable of producing damage to the biomembrane, and this biomembrane damage are capable of producing mutagenic products, especially the MDA, the malonaldehyde. This is malondialdehyde. So this is one of the assay that you use as an indicator of the oxidative stress in the cell, the MD mediated cell damage, cell membrane damage. And the ROS can both influence the cell proliferation as well as it can modulate the intracellular signaling. Then xenobiotics can increase the intracellular oxidants by multiple pathways. One is the CMB mediated pathway, the calcium calmodulin pathway, the transcriptional factors, and such pathways. And the next one is the MAP kinase pathway, then effect on gene proliferation, NF-kappa B, that is another pathway. So in this picture, it clearly describes how these changes are happening. Okay, the chemical carcinogens will activate the ROS. ROS will activate the MAP kinase, and this MAP kinase will activate the transcriptional factors, and this transcriptional factors will activate or produce cell proliferation, differentiation, and apoptosis. Then same way, NF-kappa B pathway, the chemical carcinogen, they will activate the ROS, then NF-kappa B, and this NF-kappa B undergoes two pathways. Among this is the more carcinogenic pathway, which will go and bind with the nucleus. It will cross through the nuclear membrane, and that will activate the transcription. So that will elevate the cytokine levels, then cell proliferation genes, adhesion molecules, everything will be increased, which will is a classical pathway that is happening in the progression phase of the tumorigenesis. Then the next one is the gap junction intracellular communications. The gap junctions we have already studied in the muscles as well as the cardiac muscles, the skeletal muscles, smooth muscles, and the cardiac muscles. They are formed by the connection proteins, creating a conduit between the intracellular and extracellular. So the calcium can move from so calcium or sodium or the depolarizing molecules can move from inside to outside through this pathway. And they are responsible for the normal contractive process that is happening, and they can suppress the uncontrolled cell proliferation in the normal cells. In carcinogenesis, this is the significance of the gap junctions. So they suppress the uncontrolled cell proliferation, and as communicating a gap via junction, the normal cells are synthesizing, or the normal cells are transmitting the growth inhibitor cells to the potentially abnormal cells. So when a cell tries to multiply, the adjacent cells will suppress, because if the cell is multiplying, there should be enough space for the two cells to occur at that place. So what the adjacent cell will do is, if one particular cell, a clonally selected cell, is trying to expand, the adjacent cells will inhibit the proliferation through the signals of cations through the gap junctions. Okay, now this gap junctions again is essential for the organ regeneration. In pathology, that is their significance. The regeneration of the organs such as liver, the gap junctions decrease during active regeneration and return as the normal tissue structure once the function is restored. So this is how the gaps are actually filled in, or if there is a necrosis, there will be some cell loss. Appreciable number of cells are lost. What will happen is that this cell hepatocytes should means they should undergo a fast rate of mitosis. Okay, so that is the role of gap junctions in the pathology, and it is linked to the carcinogenesis by reduced communication is observed between the cancer cell and the so as I said, when the cells are proliferating, the adjacents will inhibit the proliferation, excessive proliferation of one particular cell, because they communicate through the gap junctions. But when carcinogenesis is developing, the gap junctions undergo dysfunction. And when this undergoes dysfunction, the adjacent cells nearby, they are unable to control an uncontrolled proliferation that is happening in the tumor cell. Okay, and such loss of inhibitory cells in the normal cells permit uncontrolled growth and clonal expansion of the cell. So this is one change that is happening in the clonal expansion phase. Then the effect of tumor promoters. The tumor promoting genes, agents such as the phenobarbital, phthalate, dieldrin, 4-polyesters, they will inhibit the gap junction communications. And when this gap junction communication is inhibited, what happens is that the clonally selected cell will start expanding uncontrollably, and that will lead to the development of the carcinogenesis. Okay, and then inhibition is tissue and species specific. So this inhibition, say phenobarbital, can produce these changes in case of rat, but it may not be able to produce a change in the mice. So such species specific variations, it can produce dysfunction of the gap junction in the liver, but it may not be able to produce any such changes that is happening in the kidney. So that is what is known as the target organ and the species sensitivity. Okay, and it is not observed in case of non-target tissues. Say, for example, the skin cells or bone cells. Such phenomena is not there. The gap junctions are not that much obvious in those cells. So the mechanisms of carcinogenesis mediated through the gap junction do not exist, or they do not have any significant role in case of such tissues. Then the proposed mechanisms of inhibition is the decreased synthesis or increased degradation of the gap junction protein. So remember, these things are not clearly given in the textbook. They have given some bits and pieces only. So I have referred in some other textbook to create this material for easy understanding of you. Then modifiers of the chemical carcinogenic effect. Certain agents are capable of modifying the already existing carcinogenic agents. So most probably they will be the enzymes. So one specific enzyme that is present in a particular tissue is capable of converting a pro-carcinogen to a carcinogen. Okay, because the carcinogen metabolizing enzyme, or the agents which are capable of converting the pro-carcinogen to carcinogen, will be tissue specific, and they will vary from cell type and subcellular location, and it will be differently expressed across the species. So these two are the reason why there will be variation, intra-species variations. Whereas this is the reason why there is inter-species variation. Okay. Then inducibility of carcinogenic metabolic enzymes. And many of such enzymes are induced by many of the carcinogenic agents. The one of the mechanism by which they act as a carcinogen is that because they themselves require to be activated to from the stage of procarcinogen to carcinogen. So what they will do is they will activate the enzymes that are capable of converting the procarcinogenic agent to carcinogenic agent. Okay. That is when we studied about the benzopyrene, we already studied these mechanisms. Then additional inducers include the drug metabolizing enzyme like the phenobarbital, the cytochrome P450, which is responsible for the metabolism of phenobarbital, DDT, ethanol, 17-beta estradiol, PPR alpha agonist. All this we have studied when we studied about the AHR and other receptors. Okay. Some carcinogens are capable of inhibiting the drug metabolism. So previously we have seen the activation. Now this is the inhibition of the drug metabolizing enzyme, and that will lead to an increased half-life of the carcinogen. So they get more time to act as a promoting tumor promoter or the tumor propagators. Okay, the next one is genetic polymorphism and SNPs. When we studied about the DNAs, we have seen the single nucleotide polymorphism. Generally, the SNPs do not produce any carcinogenicity. The chances are very, very rare. But you can see so many SNPs because SNP is one that is responsible for the intra-species variation in the. So it is a part of the evolution of DNA. Okay, so such evolutions sometimes will have a setback of uh, making certain persons more sensitive to the carcinogens. Okay, in some persons, even a very minute exposure to the carcinogen can develop into a tumor, whereas some are more tolerant. And the SNPs or the single nucleotide polymorphism. Say SNP means one nucleotide is different from other persons. Only one nucleotide, maybe of an essential gene or any other thing, which may or may not affect the activity of the DNA. Sorry, that particular enzyme. Okay, but unfortunately, in such persons, in some persons, very rarely that happens. In some persons, they will become more sensitive because the enzyme will not be optimally functioning. The drug metabolizing enzyme or the carcinogen xenobiotic metabolizing enzyme may not be properly functioning. So when you study about the fast accelerators and slow accelerators in the as the racial variation that is happening in the drug metabolism, this is one of the example where the SNPs are responsible for the are partially responsible for the fast acceleration and slow acceleration. So the same thing can happen with any of the xenobiotic or even a carcinogenic agent. Then glutathione transferase polymorphism and cancer risk. So GST polymerization is also responsible for, or GSTM null is linked with a 1.5 times higher bladder cancer risk, 1.7 times gastric cancer risk, then 3 times GST-T1 null genotype. Again, the polymorphism that is happening in the GST. Times higher breast cancer, 1.6 times the gastric cancers, 4 times the liver cancers can happen. So not only the drug metabolizing enzyme, even a simple enzymes like the glutathione transferase polymorphism can lead to the development of or making the person more susceptible to cancer. They will not produce really the cancer directly, but they make the persons susceptible to the cancer. Okay, so these are the another variations. So the bioactivation and DNA repair genes variation. The DNA repair, we have already studied the enzymes that are involved or the pathways that are involved in the DNA repair. This also can produce or make the persons more or less susceptible to the. So these are the individual variation that is happening within the species. Okay, this is not across the species. This is within the species. Now, inorganic carcinogens. So somehow this topic is apparently kept in the CND in the carcinogens because we study about these metals in detail when we study about the metallic toxicity. So whatever that is given in the CND textbook, I have given it separately, but I'm not going into it because you have already studied the toxicity of these metals, including the carcinogenesis. Anyway, it will be there in the slide. And I think one thing that may not be mentioned along with the metals will be the beryllium. Usually the beryllium exposure occurs to a person at workplace, and other sources like the inhalation of beryllium dust, then tobacco smoke, then ingestion of fruits, vegetables, etc. Can lead to the intake of beryllium into the body. And the beryllium act as a negative or weak positive result in mutagenic test. It is not a full carcinogen, but they are capable of producing because the beryllium is a very small metal. small metallic ion. As a atomic number is 4. It is having only 4 electrons. So they are capable of releasing the electrons easily, the outer 2 electrons easily. So they can bind with the nucleoproteins. They can inhibit the enzymes involved in the DNA synthesis, can cause infidelity in DNA synthesis, and can induce gene mutations. All this are the common class means mechanism of the metallic carcinogens. Then animal studies have shown that this rodents, it is toxic. It is producing lung tumors in non-human primates and rodents. Then tumor-inducing compounds. The specific findings include the anaplastic carcinoma in monkey, osteosarcoma in rabbit. But clearly, that whether it is applicable to the human beings is still a debate. Okay, coming to the various classification and epidemiological evidences are also there, but many studies have two types of activities. Means some say that it is having carcinogenic effect, and some studies found there is no direct relationship between the beryllium and cancer risk. So it's a questionable compound, but still, when there is a suspicion, unlike any other toxicity, the carcinogenicity should be given a very high priority. Then also cadmium, chromium, nickel, lead. It has been already covered. Now we will go to another important portion, that is the assessment of carcinogenicity, which that are used for assessing the carcinogenicity. So this forms the last part of the carcinogenicity or the chemical carcinogenesis in C&D. Now, there are three tests. One is the short-term test, and medium-term test, and the long-term test. Short-term test actually is done in case of the cell lines, usually done in the cell cultures. It can be up to several weeks to months. And the medium, it is a qualitative and quantitative analysis of the pre-neoplastic conditions, which will take 2 to 8 months. And the long-term study will include 18 months to 24 months. That is 1.5 year to 3 year study will be the one that is classifying as the long-term study. And usually the long-term or the chronic study is done in case of animals. And the short-term test for the mutagenicity, the purpose is to designate it to find out, identify a potential carcinogenic amount by chemical by their ability to induce DNA mutations. And they are used as a surrogate test for the carcinogenicity, and the direct carcinogenic toxicity it is less effective for non-genotoxic ones. Okay, so this will work with the genotoxic carcinogens, but it may not work well with the non-genotoxic ones. Okay, because basically these tests are done to evaluate the genotoxicity rather than direct carcinogenicity. So indirect carcinogens, which are not acting through the gene modifications, we have already discussed such agents, they may not be detected by short-term test for mutagenicity. So this is one of the drawback of this test. Then in vitro gene mutation assays, the classical example is the Ames test that all of you will be knowing. The Ames test, I have given in detail about this test. So what we do is we'll inject the inducers of the hepatocellular, that means the liver enzymes. After 1 week, the liver will be collected, homogenized, centrifuged, and the supernatant, the S9 fraction will be collected, and it will be mixed with the bacteria to the test chemical. So what happens is that when we add the carcinogen into this, it will be converted to the pre-carcinogen will be converted to the carcinogen, and then we add the bacteria, do the regular Ames test. So the Ames positive, when you get the revertant, that is the histidine plus colonies are observed, that indicate that the substance that you have used is a pro-carcinogenic agent. That means once, even if it is giving a negative result with the classical Ames test, when we inject it into the animal, and when the pro-carcinogen is converted to the carcinogen, it can become carcinogenic. Okay, so it does the indirect carcinogens also. And the next one is in vitro gene mutation assay. Generally, we use the mouse lymphoma and the CHO cell assay. So these are the classical tests that are done to do this. So what we do is we typically conduct without metabolic activation. Usually the S9 inactive means non-activated S9 fraction will be collected. The drug will be mixed with that one, or the chemical will be mixed with that one, and we will do the assay with the mouse lymphoma cells or the CHO cell. Now, uh, HCPRT gene assay. Again, this is described in the textbook. Passively, they have mentioned in the CND. That is why I have given, because many of the people who are attempting the exam may not be knowing the significance of the HCPRT. That's why I have included this slide also. This is actually not from the CND textbook. Actually encodes the HCPRT, which is essential for the purine salvage pathway. That is the hypoxanthine pathway, and the recycling of the the DNA bases are happening with this pathway, and it helps to maintain the normal nucleotide pool for the DNA and RNA synthesis, and the deficiency can lead to. So this, we are not much concerned about the carcinogenicity. Just I am mentioning what is a normal physiology. Now, when the mutation is happening, there will be deficiency of the purine analogs, and that will make the compound more toxic to the nucleotide analogs of the purine. So when we apply a nucleotide analog as a carcinogen, when a nucleotide analog is acting as a carcinogen, what happens is that the chance of binding it with the DNA depend on how much purine, normal purine bases are present, and how much the aberrant purine or the nucleotide analogs are present. So what happens is that when this gene misfunctions or malfunctions, the level of innate purine within the cell will be very, very less. Because of that, more of thioguanine or the nucleotide analog will be binding with the DNA, which will produce instability. And when that instability comes in the tumor suppressor gene, the tumor suppressor gene will be inactivated, leading to the initiation of tumor. Then the mammalian cells that we already studied, the mouse lymphoma cells, HCRT, CHO cells, this is one principle, means a test that we do for the HCPRT test. The assay, the loss of HCPRT phenotype through the mutation prevents the purine analog incorporation, and the mutant cells will survive exposure to the toxic analogs. And it can be performed with the presence of an activation system or a hepatic feeder layer. So there will be metabolic activation. The precarcinogen also can be active or tested for the activity of carcinogenesis by this method. Now, the next one is the thymidylate kinase enzyme. So thymidylate kinase again is responsible for the somatic gene involved in the pyrimidine metabolism, just like the purine metabolism. In case of HBGRT, this TK mutation assay is related with the pyrimidine base. Okay, so TK positive or cell negative cells will be treated with the cell, and the fluorouracil is the compound that will be used. So in case of the previous one, it was the thioguanine. Here it is fluorouracil. So if the mutation TK occurs, the fluorouracil will not be metabolized, and only the mutant colonies are capable of surviving. Okay, so this is the principle of the test. After exposure, the cell will be incubated for a recovery period, allowing for the mutation fixation. And TK and HBC, the trifluoromethylene, and for HCPRT, we will be using thioguanine. Okay, and we'll have a scoring of the mutant colonies, then cytotoxicity, the data interpretation, and the regulatory use of this test. So this is not actually clearly mentioned in the CND textbook. This I have taken from the standard protocol for the TK and HCPRT, because unless you don't understand this, it will be very difficult to learn what is this, and in-depth questions will be difficult to answer without understanding what is the mechanism of this test. Okay, so this is the pictorial representation of the previous slides. Now, in vivo gene mutation assay, it involves the advantages over the in vitro uh, and the transgenic rodent assays. The two important transgenic rodent assays, the mutamouse and big blue mouse, and it contain a delta DNA with a LAC-Z gene, whereas the big blue will contain a LAC-I gene, which is the repressor of the LAC-Z gene. So they are having the activatory and the inhibitory indicator uh, systems. And the next one is a pig gene mutation assay. Pig gene is actually, so don't confuse it with the pig or guinea pig. It is an X-linked gene for the GPA anchor synthesis. And mutation is detected by the loss of GPA anchor surface markers. So usually it is done with the rat blood cells, RBC, or the reticulocytes. And it is analyzed via flow cytometry. And you can add specific antigens for the GPA anchored surface markers. The surface CD95, CD59 antibodies are added, and we will be sorting the cells which are positive for that one and negative for that. So mutations, when the mutation happens, the surface protein, so the surface marker, the CD59 will be absent in those cells. Okay, due to the carcinogenic effect of the compound. And why this is very important is because it is having cross species comparison. So it is not only applicable to the rodent, it can be applicable from rodents to even the human beings, because it is a highly conserved gene. This pig gene is a highly conserved gene in all the species in the mammals. Okay, so this is the protocol for the assay. So when we use the GPA positive, the CDA positive cells will be detected as fluorescent, and the one that is mutated, or the one that is capable of mutating such a gene, a chemical that is capable of mutating such a gene, will be appearing as non-fluorescent. So the non-fluorescent levels are enhanced, means the compound is capable of producing carcinogenicity. So this is the basic principle of the rat RBC pig gene mutation assay. So don't confuse it, because even though the gene is named like our HGRT gene, it is a little bit means it's a funny named gene, even though that is present in the rat RBC. So these are the short-term assays, various assays. This is a table as such that is given in the assay in the CND textbook, and most of this we have already discussed. Then the chromosomal alterations. The chromosomal aberrations are frequent in malignant neoplasms, which will be useful in carcinogenicity testing. And assays both in vivo and in vitro are available. In vitro, we will be using the mammalian cells. Just gene sustain will be capable of identifying the karyotypes. So what we generally do is, in case of chromosomal assay, we do generally the karyotyping. We'll be visualizing the chromosome at the or the metaphase chromosome during the cell divisions, and we see whether there are 23 sets of chromosomes present there. If there is any gross change in the chromosome, like the break in the chromosome, then deletion, rearrangement, translocations, despiralization, everything will be counted. So the gene, even though the gene may not be affected, the chromosome itself will be affected. Say what happens is that a chromosome like this may be split. So it will be appearing like this, and a fragment will be there. So this is what we study in the chromosomal alteration studies. So this test is actually done with the karyotyping, and even fluorescent in situ hybridization. The fish technique also employs this to find out this techniques. Then the chromosomal alterations like the sister chromatid exchange, in vivo micronucleus test. Micronucleus is again when there will be a fragment or the small portion of the nucleus. Generally, the nucleus will be seen as a thing. So you will get an additional small fragment which may look like a nucleolus outside the DNA. So that is one classical micronucleus assay we do. And again, this is having diagnostic significance also. The micronucleus will be more in case of the tumor cells, because even before the complete division of the DNA, the cell will start dividing. So there will be incomplete division, and this incomplete division will lead to the development of fragments or the micronucleus formations. So likewise is the case with the comet assay also. So you'll get a series of nucleus or smears in the DNA or the nuclear material. Okay. First one is the primary DNA damage. And the second one is the unscheduled DNA synthesis. Actually, this unscheduled DNA synthesis measures the DNA repair increase followed by an increase in the lesion. That is, after the induction has happened, the tumor is clonally selected. When we do the treatment, how much will be reducing? Okay, that is also an indicator of the DNA damage. Then DNA strand break detection includes the S2AX study, which is done in vivo as well as after treating in case of animals in case of in vivo test. The comet assay. Comet assay also I have already mentioned. When we do the DNA assay or when we spread the DNA in electrophoresis assay, you will see a comet-like appearance indicating fragments of DNA. So the smaller fragments will move faster in case of in the gel when we run it, polyacrylamide gel when we run it, and the bigger chunks will be remaining. So it will be moving or it will give an appearance. So when we take the photo with the ethidium bromide or acridine orange, you will get a clear comet-like appearance. The tail of the comet will be formed by the fragments of the nucleus, and the bulk nucleus will give the center of the comet. It can be done in both cases, that is in case of in vivo as well as in case of cell cultures, and also in case of in vivo animals. And it is incorporated, this in vivo comet assay is incorporated into the ICH 2SR. So the guidelines I will explain, I have explained here. These guidelines are not actually mentioned in the Cassett and Dowd textbook. So I have given it separately from the actual source of the OECD test guideline. So this is based on the OECD test guideline. So these are the in brief about this particular test guideline for 89, because in the DABT exams there will be questions regarding the guidelines also. That is why I have included these guidelines in the carcinogens, because these guidelines are important or significant in case of carcinogenicity. Apart from the 253, 252 guidelines, these guidelines are also very important. Then ICHS2 guideline. Okay, you have two options: the reverse mutation assay and two in vitro assays will be there for the option 2. In option 1, first will be the Ames test, followed by the chromosomal aberration or micronucleus test, followed by in vivo test for the chromosomal damage for the micronucleus or the chromosomal aberration test. So these two options are there. And the next one is short-term test, that is the transformation assay. In vitro methods, the carcinogenic potential can be assessed using the cell lines. These are the commonly employed cell lines. So this is called as Syrian hamster embryo or SHE analysis. That is the diploid cell assay based on the transformations. And the advantage of this SHE assay is that it is having almost 85% correlation with the 2-year rodent bioassay, and the transformation stages mirror the classical carcinogenesis. That is, there will be cells which are in the initiation state, there will be cells in the promotion state, and there will be cells in the progression state. Okay, and it is capable of, especially in vivo test, is capable of finding out the genotoxic and non-genotoxic classes. Now, the regulatory reference SHE assays, a regulatory approved study for and the chronic 2-year rodent assay protocol. And I have given the brief of the protocol because in CND textbook it is given in a more complex pathway. It includes everything that is mentioned in the textbook, but in a more systematic way. Okay, the dosage monitoring, everything is there. Then organ specific bioassays. So the previous ones are the general carcinogen disease studies. Next one is the organ specific bioassay. That is particularly the whether it is having any hepatotoxicity assay, hepatostudy, toxicity potential, nephrotoxicity potential, or any other toxicity, lung, liver, and such toxicities are there for a particular organ system. That is what is being studied in the organ specific bioassay. In case of liver carcinogenesis model, the rationale is liver is a major carcinogenic target. And 50% of the NTP testing I will mention later the principles because here in the textbook it is given as a very small sentence only about that one. NTP I will tell you in detail. I will give you the PPT in detail. The stages will include the initiation, pre-neoplastic and the neoplastic endpoints. Okay, the number of cells of the size of the pre-neoplastic cells and which can be confirmed by the H&E staining or any biochemical markers also can be used. Then initiation promotion approach for this particular test involves the test is given before a promoting agent like the phenobarbital. Phenobarbital is capable of inducing or converting the procarcinogen to carcinogen. So generally it is given as a single dose. Sometimes if required, it will be giving its multiple dose to confirm once it is non-toxic with the single dose. Then promotion assay. First, what we'll be doing is we will be initially giving the promoting agent. And after giving the promoting agent, then we will be using a specific genotoxic carcinogen. Usually the used agent is the diethyl nitrosamine. Why we use the diethyl nitrosamine, that will come to that later. The next one is ITO assay, which is a standard protocol as followed in the Japan. Then duration is 8 weeks. Then usually we use the diethyl nitrosamine dose for initiation. Then 2 weeks recovery, 8-week chemical exposure, and after the exposure, 2/3 of the partial hepatectomy done to stimulate the cell proliferation to see whether there is any cell proliferation. Then endpoint will be the GST positive liver lesions, glutathione transferase positive lesions. Modified then modified ITO protocols allows the detection of the promoting agents. The next model for the liver carcinogenesis is the newborn mouse liver carcinogen. You can test the initiation as well as promotion as the endpoint. Then single diethyl nitrosamine will be used to dose the infant for the initiation. Weeks after that, DEN will be using the carcinogen. Okay, and DEN is actually considered as the standard initiating agent. Why? Because DEN is acting as a perfect spark for the liver specific cancer models. Again, these things are not there in the CND textbook, but without understanding this, it will be very difficult to understand this hepatocarcinogenicity. Why the DEN is specifically used is because it is highly specific to the liver. It generally do not act as a promotion agent, or the activating agent in the in any other organ, because the liver contain the this particular cytochrome enzyme. Okay, so no other organ can convert it into a carcinogen from a procarcinogen level, and it is having strong genotoxicity potential because even a single dose is capable of producing or alkylating the DNA. We already studied how the alkylation produces carcinogenicity in the hepatocyte and reproducibility across the species and strains. Okay, then rapid induction, well-characterized histology, control for selective initiation. All these are the advantages of the diethyl nitrosamine. Then newborn mouse liver model we have already studied in detail, but this I have given in more systematic way so that it will be easy for you to understand. Okay, so the next organ is the skin. Carcinogenicity testing in the skin actually this is also very important because the tumor development. The advantage with this model is that the tumor development can be monitored visually over time. And the tumor number and size increase in the size can be visually quantified, and it will be very easy to quantify just measuring the lesion outside the body. Even you can see the progress every day, every minute, like that you can see the progression, and is well suited for dissecting mechanisms for dissecting the mechanisms of multi-stage carcinogenesis, because it is very easy to collect the samples from the skin other than taking the biopsy or FNAC in case of the animal model, the cancers which are situated inside the body. My slide visible?
Yes, it is visible, sir.
Mhm. Oh, okay. So the two-stage concept in case of the skin carcinogenesis model is the initiation stage and the proliferation stage. Actually, the initiation actually studies the DMBA. DMBA is the most commonly used, or the UV light model, or benzoyperin. Benzoyperin is actually the first model of carcinogenicity where we started learning about a disease condition like cancer in case of the chimney suit. Then it lead to a permanent DNA mutation that is happening. And the sample collection, as I already mentioned, is very easy also. The initiated cells will morphologically appear normal, but they will retain the proliferative potential. Whereas when it is moving to the promotional stage, it involves the clonal expansion, as we have already seen. So that promotion you can very easily distinguish by the initiated keratinocytes, because the discoloration that is occurring at the skin lesion. So it is very easy to demarcate the initiation and the promotion stage. Okay, so then the progression, the third stage, the third stage, you can visually see the size of the lesion that is increasing. So generally, only 5% of the papillomas, the wart lesions, they will pro promote to, or they will progress to carcinoma. So generally, the skin lesions or the skin tumors will be benign in nature, but when we apply the compounds like the DMBT, it will develop into carcinoma, the aggressive or the malignant form of the tumor. Okay, so it can be used as an assess to assess the initiating and promoting activity. So don't think that this assay is exclusively used for the dermal carcinogenesis. So this is used for the systemic carcinogenesis also, because this is a model which can very easily you can identify the initiation, propagation, the initiation, promotion and propagation stages, progression stages. Okay, and usually the hairless mice and the Sengar mice are the one that is having high sensitivity to the skin tumor induction because of the lack of hair. Then TPA, that is the tetradecanoyl forbol, is a common forbol ester that is associated from the euphoria plants. It is not a direct carcinogen. It will structurally mimic the DAG, diacylglycerol, which will persistently activate the protein kinase C. So the protein kinase C will phosphorylate the proteins and increase the cellular metabolisms. So it will act as a tumor promoter. But since it is activating through the protein kinase, it is not acting on the DNA. So it is not a genotoxic compound. And in case of mouse two-stage model, it is applied after initiation to expand the initiated colonies. The clonal expansion is studied after the initiation. So this is the schematic of the experiment. This is as given in the CND. And carcinogenicity testing in case of lung lesions. So generally we use the strain A mouse model where the genetic strain A mice is naturally developed lung tumors. The incident rate almost 90% by 24th month. So the tumor progression, hyperplastic lesions, like the lung adenocarcinoma and the carcinoma within the adenoma, and that will be developing into the carcinoma. So the hyperplastic, so initially there will be hyperplasia. Hyperplasia will develop to adenoma. Adenoma will produce the initiated cells. The adenoma cells. Adenoma is generally a benign tumor. So after some time, you will see the adenocarcinoma cells start developing. So that is actually indicating the initiation of the tumor. Okay, and later that will progress to a carcinoma. So this is seen as a cascade or as a chain. You can see with this strain A mouse. That is the advantage of using strain A mouse as a lung carcinogenicity approach. And usually the chemical exposure is done for 8 weeks, followed by 4 months without the exposure for the observation period. So generally it is given as inhalation. So you can just check the inhalant carcinogenicity assay also with this one. And again, spontaneously some animals can develop the tumors. So you need to compare it with the control animals also. So control animal is also mandatory to assess this assay. Then other organs. Other organs very rarely we use. The kidney, bladder, pancreas, tumor, colon, small intestine, and oral cavity. The use is very much limited. Okay. Now, these are the target organ models for studying other toxicities: kidney, bladder, pancreas, stomach. Especially the stomach, two types of are used. One is a false stomach, and second one is the glandular stomach, the muscular stomach and the glandular stomach. Then colon, small intestine, oral cavity, tongue, buccal cells, the squamous cell carcinoma in the buccal cells. These are the models that is, so this is the table that is given in the CND textbook. Then transgenic animals in carcinogenic assessment, the rationale and use will be the genetically modified animal increases susceptibility of the chemical induced carcinogenesis. Okay, they are not actually the ones that is having a cancer, but their sensitivity to the cancer is very high. Okay, so the common model is the TGAC model, RISH2 transgenic animals, P53 positive and negative, that is mutant cells are there, XPN knockout mouses are used, and the evaluation and finding with the HSI, ILSI reviews. This is as given in the textbook. I'll tell you in detail about this guidelines later. And could substitute mouse 2-year bioassay in some cases with the combined effect on the genotoxicity, structural activity, and other bioassay, toxicokinetics, and mechanism. So the duration can be reduced because of their high sensitivity or high susceptibility for the tumor development. The duration can be reduced to 6 months or 3 months like that, which will be equivalent to the 2-year data that is present in the that is normally used for the 2-year bioassay for the carcinogenesis. Then the limitations actually was that even though we can technically say that 2-year bioassay can be substituted with this one, but the results are not that much correlated with the or with the reduction in the time, but it is suited for specific pathway mechanisms or in the carcinogenesis. You can use it for the studying specific pathways. So for example, when one pathway is not present in a particular animal, a mutant animal, so whether the carcinogen is acting through that pathway. So if it is acting through that pathway means if that pathway is absent, the animal will not develop carcinogenesis. So for rather than using it for the regulatory purpose, it is more commonly employed for the mechanistic carcinogenesis study in case of the transgenic animals. Then the new approaches in the carcinogenicity assay as given in the textbook as the uh, motivation for the change because the traditional bioassays are time consuming and may lack human relevance. Even when we do the rodents, that may not be as good replicated in case of human beings. And the advances in the cellular molecular biology and high throughput and computational methods enable fast and more targeted. And the emerging strategies include the high throughput in vitro bioassays, in silico modeling, then microphysiological systems like the organ on the chip models. These are the emerging strategies. These things are as given in the Castret and Dowell textbook. And the major in silico processes involve the USEPA ToxCast, that is toxicological prediction softwares. Then microphysiological systems, that organ on the chip systems, are the ones that are emerging for the toxicity. Still, it is in the developing stage only. Full correlation studies have not been done. In some cases, it is working better, but how far the results are reliable in case of human beings is still to be estimated. Then coming to the human carcinogenesis, the carcinogenesis factors associated with the lifestyle. So these are the things that is given in the textbook itself. So occupational human carcinogens. Uh, these are the list of occupational human carcinogens as given in the C&D textbook. Then human carcinogen chemicals associated with the medical therapy and diagnosis. These are the list of compounds. Then chemical carcinogens in human beings, that IRC reviews volume 1A just include identified 120 agents as carcinogenic humans, and 6 categories like pharmaceutical, biological agents, metals, radiation, personal habits and household exposure, and the chemical agents are related to occupations. So these are the 6 categories in which the 120 agents are being included. Now, major contributing factors to the carcinogenesis as a lifestyle is the major carcinogen. So remember the first slide that I have given. One is the in which a pie diagram is given with the lifestyle is as the most important contributor for the, especially the food is the one that is contributing maximally to the development of the cancer. Okay, it includes the tobacco, poor diet, mold contamination, aspergillus, which will produce aflatoxin, which is a carcinogen. Then cooking methods can produce pH, then array hydrocarbon agents and such things. Then diet and nutrition. In diet and nutrition, you may expect in all other diseases, we expect that there will be an increase in the disease occurrence with a poor quality diet or poor quality or poor quantity. But in case of tumors, the overnutrition and high fat diet is the one that is contributing to an increased risk for the cancer. And then vitamin deficiencies like the vitamin A and vitamin E. Vitamin E is obvious because of its antioxidant activity. It can promote the cancer development. Then occupational exposures, the pH in chimney soil, because that was the first cancer that was being detected, or that is how the etiology is the first cancer where the etiology is found out. Then the drugs and medical treatments. Then coming to the evaluation of carcinogenicity, this is very important. This IARC classification systems, how the chemical is classified according to the IARC class. The IARC stands for the International Agency for Research on Cancer, because this IARC is coming at bits and pieces in case of the C&D textbook, but I consolidated it into a single one here. Okay, they have given us a table also. So generally, the group 1 include the carcinogenic to humans, which is having definitive evidence. Okay, 120 agents, but it is not 120 actually. Now the number has been increased. This is as per the last version of the means the C&D textbook that is given to us. Then group 2A is probably carcinogenic to compound. The 2B is possibly carcinogenic. So there will be a difference between 2A and 2B that I will mention later. The group 3 is not classifiable as carcinogenicity in humans because it is not evidentially proven that it can produce carcinogenicity. Then group 4 include probably it is not carcinogenic to human. So this is based on the epidemiological data, experimental animals, and in vitro data. Combining all those things, this classification or this grouping has been made. Then the next one is the NTP classification, the National Toxicology Program classification, part of the US Department of Health, and based on the toxicological and mechanistic studies. That is, their groups are the known to be human carcinogen, that is a proven carcinogen. Sufficient studies or evidences are there from the human studies itself. Then the second class is the reasonably anticipated to be human carcinogen. The third class is the limited evidence as human carcinogen. Then fourth class is the sufficient evidence in animals. And the fifth last class is the strong mechanistic evidence in human. So based on the mechanism, there are every possibility, because in particular human being that particular enzyme is there, and because of that there will be high chances of developing into tumor. So that is what is in the strong mechanistic evidence in the human beings. Okay, so this is IARC classification as given in the textbook. And I mentioned the two terms. One is likely and second one is suggestive, that is 2A and 2B. So likely means strong consistent animal evidence occur in multiple models and conditions regardless of the human evidence. And suggestive evidence, actually some signs, means it is not fully confirmed, but still the more work has to be done to classify it as a likely or a potentially class 1 group. So that is what is meant by the difference in the 2A and 2B. So that is why I specifically given it with the likely, or what is actually meant by the 2A and 2B. Okay, so this is how it is represented in table. So this is 2A. The first one is 2A. This is 2A, and this is 2B. So these are the tabular representation of the difference between these two. And other regulatory frameworks include the US EPA, FDA. In case of US EPA 2005 risk assessment include the carcinogenic to human, likely carcinogenic, suggestive evidence, and inadequate information about the assess the carcinogenic potential, and not likely to be a carcinogenic to the humans. So these are the classifications based on the. So you have the IARC classification, you have the NTP classification, and you have the third classification as the US EPA classification. Then this is the mode of action framework for the US EPA. How it is being done. Again, this is not given in the textbook. This I have taken from the US EPA. And the mode of approach, mode of action approach for the US EPA guideline. Then the next one is adverse outcome pathway, AOP approach. It is developed by the OECD along with the US EPA, EC, and US Army. So links a molecule initiating event to the key event and adverse outcome. I will explain this, what is MIE, what is KE, and what is AO. The KE are the key events are the measurable in every step. That is key event relationships. KER is supported by the biological plausibility and empirical data. Then mode of action. The key events and processes starting from the interaction of an agent with the cell, that is with any enzyme or any other mechanistic pathway, through functional and anatomical changes. Anatomical changes means cellular damage or the DNA damage or such changes that will result in the cancer or other health endpoints. Okay, so what is molecular initiating event? That MIE is actually the first interaction between a chemical and biological. So here when I explain, it has more MIE, which will lead MIE will lead to key events and adverse outcome. So in the textbook only this has been given. So I've gone through in detail about this, and the molecular initiating event actually is the first interaction between the chemical and biological system. That is a toxin binding to the receptor, or a DNA damage that is induced by an adduct. And this actually triggers the chain of events that leading to the harm. Here the harm means it is a carcinogenesis. Then key events. Key events actually develop from the molecular initiating events. So that will constitute those signs which are empirically observed precursor steps, that is essential for or a necessary element of mode of action or biologically based marker for such element. So that means the observable, measurable biological changes occurring after MIE. So when an MIE is there, molecular initiating event is there, what are the observable effect? Molecular effect changes means what is happening in the structural changes. Now, how the structural changes is converted to the functional changes. That is what is measured in the key events, like the enzyme inhibition, cell death, DNA maturation, everything. So how when a toxin is binding with a receptor, it produces cell death. How when a toxin combined with the DNA, it produces DNA mutation. So those are the key events. Then adverse outcome. Now, what will be the outcome? What will happen when the key event is progressing to an ultimate level? So it can develop to a cancer, it can develop to an organ failure, it can develop into a reproductive toxicity. So that is what is known as adverse outcome pathway components. Means the adverse outcome and key event relationship. So it is a scientifically supported link between two KEs. That is KE. So here, if you can see, the molecular initiating event will lead to a key event 1, key event 2, and key event 3, 4, like that. And at the end, it will develop into a tumor. So imagine that in case of carcinogenicity, it is a tumor. So how this KE is leading to KE1 is leading to KE2. That is what is studied with the help of key event relationship, or how the KE1 leads to KE2, how the KE2 leads to KE3, how the KE3 leads to KE4, and how that is producing ultimately an adverse effect. That is what is studied by the KER. Okay, now the list of carcinogens, it's a big list of carcinogens that is given in the Casret and Dowd textbook. You can refer to that carcinogens, because I'm not including in that presentation because that is a big list. Now, one thing I want to emphasize is that I have covered almost 99% of the textbook. So even without reading the chapter, except these tables, without reading the chapters, just going through this presentation itself, you can completely understand 99% of the things that is given. So every important point that is given in the textbook is given in my slides also. Maybe it's a little bit exhaustive. Some 250 to 270 slides are there, but still without reading the textbook, you can understand everything. Whatever that is given in the textbook, plus some things which will help you to understand what is given in the textbook is also included in my presentation. So I think thank you all the best for this examination, and also I thank all the organizers for giving me an opportunity to take this session.